Stable and high rice yields cannot be separated from effective weed control. In comprehensive control strategies, herbicides with their high efficiency and rapid characteristics have become indispensable technical means. As of 2024, there are as many as 120 active ingredients registered for use in rice field herbicides in China. How do they precisely identify weeds and exert killing effects? The core lies in precise interference with weed physiological and biochemical processes. Understanding herbicide weed killing principles is the foundation for scientific herbicide selection and use, achieving weed control and rice protection goals. The reason herbicides can kill weeds and protect seedlings lies in their modes of action, having high selectivity and lethal interference. 1. Interfering with core life activity processes, blocking the lifeline. Propanil and symmetrin can inhibit photosynthesis. This type of herbicide acts on chloroplasts, blocking electron transport chain or energy conversion process in photosynthesis, making weeds unable to manufacture carbohydrates essential for survival, ultimately starving to death. Affected weeds commonly show chlorosis and albinism symptoms. Glyphosate can inhibit amino acid synthesis. Amino acids are building blocks for protein synthesis. This type of herbicide precisely inhibits specific enzymes in key amino acid biosynthesis pathways within weeds. Protein synthesis is blocked, causing weed growth stagnation, deformity, ultimately overall death. 2. 4-D and quinclorac interfere with hormone balance and cell division. This type of herbicide mimics or interferes with plant natural hormones like auxin and cytokinin. They cause abnormal swelling and twisting at weed growing points, leaf deformity, vascular tissue blockage, destroying normal cell division and elongation processes, ultimately causing weeds to die from uncontrolled growth. Lipids are main components of cell membranes. Some herbicides can inhibit biosynthesis of fatty acids, waxes, and other lipid substances, causing newly formed tissues, especially shoot cell membranes, to be underdeveloped or functionally damaged, making weeds lose water, become fragile, and easily damaged. 2. Herbicides can kill weeds while not harming or minimally harming rice, mainly relying on the following selectivity mechanisms. Positional selectivity, utilizing differences in depth distribution of crop and weed root systems or coleoptiles in soil. For example, transplanted rice roots are deeper, while weed seeds mostly germinate at shallow surface. After applying soil pre-emergence herbicides, they form herbicide film in topsoil layer, can effectively kill shallow germinating weed shoots, while deep rice roots are relatively safe. Temporal selectivity, utilizing time differences in crop and weed germination and growth. After rice transplanting and recovery, when weeds have not yet germinated or are in sensitive tender stage, apply herbicide can effectively kill weeds while having minimal impact on already robust growing rice. Direct-seeded rice field simultaneous sowing and spraying technology also utilizes rice seeds' detoxification ability, while weed shoots are highly sensitive. This temporal difference, morphological and physiological selectivity, Differences in morphological structure and physiological and biochemical processes between crops and weeds. For example, poaceae herbicide Cephalophore butyle can be efficiently absorbed by sensitive weeds like barnyard grass, translocated to growing points, while rice can rapidly metabolize it into non-toxic substances, thus safe for rice. Detoxifying enzymes contained in rice are important physiological foundation. 3. Mode of action and targets. Systemic type like glyphosate, benzofuran, methyl. After herbicide is absorbed by weed roots, stems, leaves can move upward or downward in xylem and phloem, translocating to whole plant, achieving thorough eradication effect. This type of herbicide is suitable for controlling perennial weeds or large-age weeds. Contact type like propanil, paraquat. Herbicide mainly acts on parts contacting herbicide solution, causing local tissue necrosis and desiccation. This type of herbicide takes effect quickly, but difficult to translocate to non-contact parts. Poor effect on underground parts of perennial weeds. Commonly used for rapid post-emergence, early killing, or as foliar-applied herbicide. According to field dominant weed species and their growth stages, select herbicides with corresponding modes of action. Based on herbicide mode of action and weed, most sensitive period for application. Missing window period reduces efficacy and easily causes crop phytotoxicity. Insufficient dosage cannot reach lethal interference concentration. Excessive amount destroys selectivity, easily harms rice seedlings, and pollutes environment. Uniform application ensures weeds fully contact herbicide or soil herbicide layer is complete. Long-term single use of herbicides with same mode of action will select resistant weed populations. Should rotate herbicides with different modes of action or scientifically mix, delaying resistance development. Temperature, light, humidity, soil conditions significantly affect herbicide absorption, translocation, and activity. For example, under high temperature and strong light, applying contact herbicides easily causes crop scorching. When soil is dry, pre-emergence effectiveness is poor. Herbicide weed killing principle is essentially utilizing their specific chemical structure to precisely interfere with weed key life processes, while relying on positional, temporal, morphological, and physiological differences to achieve selective crop protection. Deeply understanding modes of action and selectivity principles is the foundation for scientific herbicide selection, precision application, enhancing effectiveness, ensuring rice safety, and delaying resistance. We should always uphold prevention first, comprehensive control principle, viewing chemical weeding as one component in comprehensive management system, combining agronomic measures, physical and biological control, achieving rice production's green, safe, sustainable development.